Hello and welcome. I'm Nidhi Bansal, editor of Current Protocols at John Wiley & Sons Publishers, and I'm delighted to introduce today's webinar titled Single-Cell Analysis, Getting More from Your Data. This webinar is being co-sponsored by BD and Current Protocols, a Wiley publication. BD is a global medical technology company that is advancing the world of health by improving medical discovery, diagnostics, and the delivery of care. BD leads in patient and healthcare worker safety and the technologies that enable medical research and clinical laboratories. Published by Wiley, Current Protocols is in its 31st year and provides the highest quality, peer-reviewed, and regularly updated step-by-step research protocols to life scientists worldwide, with 18 titles and over 20,000 protocols containing exquisite procedural detail, extensive troubleshooting tips, and example results. Current Protocols provides researchers with reliable, efficient methods to ensure reproducible results. During today's presentation, we encourage you to submit your questions throughout the event by clicking on the Q&A box at the bottom of your screen. Your questions will not be seen by any of the other attendees. The webinar will be recorded and available for viewing in the next few days. We will send you an email with details of how to access the on-demand webinar. Now, it is my pleasure to introduce today's speaker, Dr. Mirko Kursalli. Dr. Kursalli obtained a PhD in clinical and experimental hematology and oncology from the University of Genova, Italy. He then transferred to the University of California to continue his research as a postdoctoral fellow. Most of his academic work has focused on the biology and therapeutic potential of human mesenchymal stromal stem cells. He is an R&D staff scientist at BD Biosciences, where he is a flow cytometry subject matter expert and is also involved in developing innovative immunology and immuno-oncology applications. So let's get started, and a very warm welcome to Dr. Mirko Kursalli.
Thank you, Nidhi, for your kind introduction. I want to thank you and welcome you all for joining today's webinar. I'm particularly excited to show data generated by the R&D immunology application team together with the genomic application team that was aimed to generate biologically relevant data to showcase the power of single-cell analysis when analyzing heterogeneous samples. And I will start from the cartoons they show here from a review from Ye et al., where authors really nicely showcase the power of single-cell analysis when addressing important biological information about heterogeneous samples. So if we start from a tissue or a sample that contains different cell types, so heterogeneous in nature, and a bulk analysis is performed, the readout and eventually the information that are obtained are an average of the gene expression or protein expression of all the cells that form the tissue or they're included in that tissue. And the implication of this is that information about rare yet critical components of the tissue, such as stem cells, cancer cells, will be masked, eventually lost, due to the average of all the other signals of other cells that are present in the tissue. Therefore, it's becoming more and more critical to analyze cells at the single cell level, where the information about each and every component of the original tissue is now recorded and can be analyzed regardless of their frequency or abundancy. So it really provided a very comprehensive characterization of all the components of the tissue. And today, we will focus on two single-cell analyses, such as flow cytometry and single-cell RNA sequencing. How can they be used individually to address cell heterogeneity, but most importantly, how the integration of both can really take us to a further level of characterization of cell heterogeneity. And this is important because, as I mentioned, cell heterogeneity is an inherent characteristic of most, if not all, biological systems. And as we show with bulk analysis, information about rare but still critical cell components like stem cell or cancer cells, is usually lost when bulk analyses are used for average measurements. But with advancement in single-cell technology, today we can perform a more precise characterization of all cellular components of the system. And this allows us to gain a deeper understanding of all the interaction and networks, cellular networks, and eventually the mechanisms governing normal as well as malignant cell development. Starting from flow cytometry, this cartoon depicts a scenario where we start from a heterogeneous sample and we, for example, induce differentiation, and then we want to assess differentiation by expression of certain proteins. If we perform a bulk analysis such as Western blot in the top panel, and we perform a total cell lysis, we can appreciate indeed that as compared to the undifferentiated sample, upon differentiation, these protein 1 and 2, just for example, are expressed, thus confirming that differentiation occurred in our sample. However, it would be impossible for us to determine how many of the cells responded to the stimulation and whether it's only a discrete specific subset that did so. And that's where flow cytometry can really address those questions. If you look at the bottom part of the cartoon, now the same sample, if it's dissociated and we create a single cell suspension that we run through the flow cytometer, every single cell will be interrogated for the expression of each protein. And now we can appreciate that as compared to the undifferentiated samples, a discrete percentage, in this case 57% of the cells, express both proteins. So it really tells us in a quantitative way how many cells have responded to a stimulation. Furthermore, and relevant to this webinar, the same technology can also use to isolate and purify only cells of interest for downstream analysis. So the second technology would be single-cell RNA sequencing, or as we'll refer moving forward, will be single-cell RNA-Seq. And we can draw kind of a parallel here with the comparison of bulk analysis like Western blot and flow cytometry. In this case, if we look at a sample, heterogeneous samples, and we're interested on the analysis of two rare cells, represented here by the two red dots. If we look at traditional RNA-Seq, so bulk analysis, the information of those two rare cells will be masked and overall lost by the information of all the other cells that we're not interested. So that the advancement eventually of typical single-cell RNA-Seq allowed us to basically distribute cells either in a plate or a microchip so that we can separate the cells and analyze them at the single-cell level. However, if our cell is still rare, that means that we will use a lot of sequencing for cells that we're not interested in and eventually will be discarded from our analysis. So it really becomes powerful that when you combine flow cytometry and single-cell RNA-Seq, you can have a way more efficient workflow. As depicted now at the bottom of the cartoon, where we show that we can take advantage of the ability of purifying cells of interest based on FACS sorting, and in this case specifically we use a BD FACS melody cell sorter, and then perform single-cell RNA-Seq only on the cells of interest, which will result in a greater improvement of efficiency and precision. But while these two technologies, single-cell RNA-Seq and flow cytometry, has evolved over the years in parallel as a complementary technologies, what's really becoming a very powerful tool is the combination of the two in what we call a system biology, where we can integrate biology technology with the help of computational analysis. So what we'll show today is how we can integrate flow cytometry data with genomics data and perform a deep analysis with the help of bioinformatics tools. How can we integrate flow cytometry and single-cell RNA-Seq then? So let's start from the workflow that we described before. We analyze our sample by flow cytometry. We identify a population of interest. We can now sort that population we can perform a single cell deposition into, in this case, a multi-well plate, and the cells that have been deposited in the plate, that would be taken for a downstream genomic application in single-cell RNA-Seq. However, in the absence of index sorting, the function that we are going to be talking about today, the information about each cell that was deposited in a specific well and the corresponding gene expression profile will be lost because cells after deposition may have been pooled, they've been analyzed for single-cell RNA-Seq, and we will not be able to trace back the information at the protein level for a specific cell of interest. And so index sorting was developed exactly to fill that gap, where now we can retrieve information about each cell of interest at the protein level, and we can perform a correlation of protein and gene expression profile at the single cell level. And that's going to be the core of today's webinar, and what we are really going to showcase is one of the available methods and technologies to perform a simultaneous protein and gene expression analysis. So just to reiterate what index sorting is a flow cytometric record of each cell that has been individually sorted, and the information about the given cells can be retrieved at any time post-downstream analysis. And this really enables the correlation of protein expression with gene expression profile or cellular functions. Today, we will focus specifically on gene expression analysis. And here I want to just show another example just to reinforce the concept of index sorting and how it relates to what we will be talking about today. So if we look at the first plot on the left, this is our flow cytometric analysis prior to cell sorting. And for this example, we are interested in purifying a population here in blue that express both marker A and B. So in the absence of index sorting, without index sorting, we will draw a gate, as you can see in the middle plot, sort the population of interest, and then we can do a single cell deposition for downstream gene expression analysis. However, if we identify a subset or a cluster of cells with a specific gene expression profile, we will not be able to retrieve information at the protein level and see, for example, if there was a correlation with the level expression of marker A, that, as you can appreciate here, can be expressed at a high or low level. Now, if we perform a similar experiment, in this case, a different experiment is showcased on the right, but with the same markers, all the information at the time of sort have been recorded for each individual cell. And so this means that now we are able to identify a cluster of cells with a specific gene expression analysis. We can go back to the protein data and define whether there was a correlation with the level of expression of marker A. Do they correlate with cells expressed high level or low level? And that was going to be really the power of index sorting, that we are not considering our population of interest as just one total cell defined by our gate, but we are going to be able to dissect our original population based on the information of each cell within the original gate. And for today's webinar, we're going to put the concept of integration of flow cytometry and single-cell RNA-Seq through index sorting in the context of the very well known and characterized and studied model of T-cell differentiation. Because it's very well known that the T-cell differentiation is a highly heterogeneous system, as depicted here in this cartoon from a beautiful review from Nicolas Restivo and Luca Gattinoni. And we know, it's very well known in the field, that upon antigen recognition, naive T cells begin a process of differentiation that ultimately lead to the generation of effector T cells. But throughout the process of differentiation, these cells will go through different stages. And now they've been divided into stem cell memory, central memory, and effector memory cells with unique immunophenotypic and functional features. The field has defined a set of surface markers, the combination of which can broadly define these different stages. However, it is very reasonable that even within a compartment, for example, the sensor memory, that is defined by these immunophenotypic profiles, heterogeneity can exist at, for example, at the gene expression level. And that's why we're going to use as a model to showcase how integration of flow cytometry and single-cell RNA-Seq can help the scientists to better define and refine the characterization of T cells at different stages of differentiation. So we'll briefly go through the experimental design, and we started by optimizing a multicolor flow cytometry panel for the single-cell deposition of T-cell subsets into 96-well plates. Once this was performed, we went on to perform downstream single-cell RNA-Seq. Then we analyzed differential gene expression across different T-cell subsets. And finally, we will show how we can further resolve cell heterogeneity by correlating protein and gene expression profile thanks to the index sorting feature. Starting from the panel design, we utilized best practice of panel design in order to guarantee maximum resolution and sorting precision of the population of interest by taking consideration antigen density, antigen co-expression, and fluorochrome brightness and spillover. This panel was designed for a fax melody cell sorter with a 3-laser configuration with a violet, blue, and a red laser. And it contains a specific surface marker that, as depicted in the previous cartoons, have been very well known to describe different subsets of T cells throughout differentiation, as shown in the cartoon below here. Before performing our single-cell deposition and downstream single-cell RNA-Seq, we want to validate our panel and make sure that we achieve a good resolution that allows us to set a very distinct gate that will also result in a precise cell sorting without cross-contamination. And we start from the top panel. These are plots generated on fax colors. It's the software of fax melody. And we can clearly separate and discriminate CD3 total T cells. From that gate, we can further narrow down on cytotoxic CD8 positive T cells. In the third plot, you can see the combination of CD45RO and CD197 CCR7. It's used to define effector memory T cells as CD45RO positive, CCR7 negative. The double positive cells are what are considered to be central memory T cells. And from the single positive CCR7 T cells, that's what we call as a naive live cell gate, we can go to the next plot where we have CD95 and CD183 CXCR3. And stem cell memory T cells are defined now within this gate as CD95 positive T cells. And for this experiment, we consider as naive cells that express low to no level of CXCR3. We also performed a bulk sort just to confirm that indeed all the cells that we have gated and sorted when we perform a post-sort purity check, we get very high level of purity without cross-contamination. So now we validate our panel, we validate the resolution, we validate our sorting precision. We are ready to perform the real experiment where we are repeating the assay, and now we can set up a plate. In this case, we use a 96-well plate where we are going to indicate the software where we want each cell of interest deposited in each well of the plate. And just to add a little bit more of statistical significance to our analysis, since we are sorting 4 populations, in this experiment, we sorted 3 plates. Cells were distributed in each plate just to avoid any kind of batch effect or to have a very standardized single-cell deposition, and we will perform eventually the single-cell RNA-seq on the 3 plates that were sorted. And lastly, before going to the data, I just want to give an overview of how we perform the analysis once we have finished our experiment and how we prepare for the bioinformatics. So at the end of the sort, when you use index sorting, an index sort CSV file is generated. And this is basically a spreadsheet, an Excel matrix that contains the recorded information for each deposited cell. So we record for each cell that is gone into each well, the MFI for each parameter that was used for the sorting. Once we've sorted the cells, we process the cells for library preparation. And this experiment, we use a targeted assay containing 220 T-cell specific genes prior to single-cell RNA-seq. And we will talk why we use a targeted assay for these genes. Once the sequencing is complete, we obtain a similar CSV file for the sequencing data that now contains for each cell information about the expression of all the genes. And what we're going to do, we're going to merge the index sort file that contains the information of the protein with the sequencing file that contains the information of the gene to create just one combined matrix that we can now upload in Sigic software. And we can have now in a single file all the information that we need to perform simultaneously protein and gene expression analysis. I will not go into the detail on how to perform these steps and how to prepare for the bioinformatics. I will rather focus today on the biology. But I highly encourage you to visit the FlowJo website and the FlowJo University website, where you can find very comprehensive and extensive tutorials that can guide you through this process. And here I just want to briefly show the interface of Sigic that resemble really the interface of FlowJo. And it was developed this way to really facilitate the analysis of gene expression profile and protein expression for scientists, and they've been familiar already with the workflow of flow cytometry analysis. What I've indicated here in the top red box is what we call the gene pane, and that's where we can see, for example, that in this file, we have information about the flow data. So these are the 7 fluorescent parameters that were recorded at the time of cell sorting. And we have the gene information, where we have all the 220 genes from our targeted assay. In the bottom red box is the sample pane. And what we have here is the 3 samples that were uploaded in the system. So the single CSV file from each individual plate. And you can also see that we perform a concatenation where the information from these 3 samples have been merged together into a single file, for which now we can perform analysis of both gene and protein expression simultaneously. Now, the great advantage of having index sorting, sequencing data in this platform, in the Sigic platform, is that we can perform conventional flow cytometry analysis based on protein. You can see here in the contour plot. We can do dimensionality reduction based on the genes, as you can see in the t-SNE plot. And you can also perform differential gene expression analysis, all in one platform, all from a single file. And again, I want to guide you a little bit on the workflow, how we approach now the data analysis. Now we have information about protein, we have information about genes. We can analyze them individually, but most importantly, we want to integrate them and analyze them in an integrated fashion and correlate protein gene expression. So as I mentioned, we start by concatenating this index, sort, and sequencing files from the 3 plates. We can recreate the plots following the sorting gating strategy, and this allows us to visualize all the cells that were individually sorted and set gates around those cells and define the population of interest. So we're going to define our subset, and then we can move to start looking at the genes So we look at we can define the same subset. Now we look at the genes. We have 220 genes, so we'll start from performing a principal component analysis, followed by a t-SNE analysis and t-SNE dimensional reduction. We'll then perform differential gene expression analysis. And eventually, at the very end, I will show you how we can use index sorting to further resolve cell heterogeneity by correlating protein and gene expression. So first, we're going to look at the protein data. So if we look at the top of this slide, those are the plots and those are the cells before sorting. Now we sort all these different subsets at the single cell level, and we have imported now this data into Sigic, and we have concatenated all the files. So basically, all the plots, all the dots that you see in the plots represent a single cell that came from the gate of origin. And this, on the bottom, is the visual representation from Sigic, where you can see now you have individual dots. They all represent a single cell. We can confirm that exactly we have the population of interest. But most importantly, each cell, each dot you see there, will have combined information about gene expression analysis. And we'll show you different ways that we can use that information to our advantage. But from now on, when I refer to protein analysis or protein signature, it means that we have defined the subset based on these colorimetric parameters that were recorded at the time of index sorting. And we can eventually, for example, interrogate cells within a defined gate for gene expression analysis. After looking at the protein expression, now we're going to look at the gene expression. And to facilitate the visualization of cells with differential gene expression, we are going to perform a dimensionality reduction, where we start from a principal component analysis and followed by a t-SNE analysis based on the expression of the 220 genes. And we can now reduce the information to a bidimensional plot, as you can see here, where each dot in the plot represents a cell, and the proximity of two individual cells, it's a function of the similarity of their gene expression profiles. So if you look, for example, across the x-axis, cells that are close to each other will have a similar gene expression profile. Cells that are on the opposite end, they will have a very different gene expression profile. However, we are not able at this point to determine which cell is an effector memory, a naive, or a stem cell memory. At this point, we are just performing a visual assessment of the cells. But now we can take advantage of the protein information, protein expression information that was recorded at the time of sort via index sorting to overlay this information on the t-SNE plot and define where in these bidimensional analysis each cell population falls based on the correlation of protein and gene expression analysis. If we look here, the naive cells that were defined based on the protein signature, we can see that they cluster nicely close to each other at the right end of the t-SNE plot. We then overlay the stem cell memory T cells based on their protein expression, and you can start seeing that they move to the center of the t-SNE plot, right in the middle. A similar distribution is observed for central memory. And now when we look at the effector memory, they nicely cluster all together at the opposite end of the naive T cells. So you can see here that we are combining the gene expression analysis used to create this cluster, and we overlay the protein information, and it biologically really tells us that naive T cells, they are very similar among each other, and they are very defined. Stem cell memory and central memory, they fit right in the middle, so again, suggesting a transition to eventual effector memory, that they are more homogeneous and at the opposite end of the naive T cells. Now we're going to look at the gene expression analysis of these different T cell subsets. And as I mentioned before, we use here a targeted approach instead of a whole transcriptome analysis because we know what genes are expressed by this subset, and we want to focus with high resolution and high sensitivity only on these genes. So in this case, we're going to refer to the seminal paper that was published by Recifo, Mario Roederer, and Luca Cattinoni's team, where they defined for the first time this human memory T cell subset of the stem cell memory T cells. And they showed, as you can see in this slide, naive associated genes and effector associated genes. They are progressively either downregulated or upregulated. And you can see at the bottom that they are progressively regulated going from naive to stem cell memory, central memory, effector memory. So we're going to use these same genes, and in this case, in this paper, the analysis was performed at the bulk level. So we're going to use these genes as a control for our system, but further we're going to look at these at the single cell level and even more in correlation with protein expression. So one quick way to perform a control in this case using these known genes, we can correlate protein genes by overlay again. So on the top left, you can see that that's the protein profile, and we can define, for simplicity here, I will only show effector memory, central memory, and naive-like cells. And now we can use a statistical heat map function that it will keep on the background the protein signature, and then we can select the gene of interest, and it will color code the cells that express the gene based on the level of expression. For example, if we look at left one, which is a naive associated gene, you can see that you see green and red cells, so positive cells, in the naive-like cells and the central memory, but not in the effector. And notably, if you look at effector-associated genes, for example, granzyme A, you can see that the cells that express these genes overlay with cells express a protein signature corresponding to effector memory indeed, but not on naive cells. So this is a very quick and easy way to confirm the pattern of expression of genes and proteins. But what also this information tells us is that if you look at granzyme A, this is homogeneously pretty much expressed by effector memory cells. Since we're looking at one cell after one cell at a time, you can see some genes, like hyomes, for example, that's expressed by a subset of effector memory. So this is already telling us that even within those gates and those cells that we conventionally define as effector memory, there is heterogeneity at the level of gene expression profile. And so for a more comprehensive analysis, now we're going to move to the analysis of the differential expression of 220 genes. And again, I would like to spend a little bit of time here just to show you, it's a busy slide, but it's a very powerful slide, and it's really the core of what we're doing here and showing today. On the left side, again, we have our protein data. So we set the gate based on the signature, and we can identify our subsets. Now we can interrogate each population within the gate for the expression of the 220 genes. So on the right-hand side, you can see that we have the RNA expression data analysis. And each column corresponds to the population of interest defined based on the protein expression, defined on the immunophenotype. And we can start observing some very interesting data. First of all, if we look at left one, for example, at the top of the list, and the blue arrow represents naive associated genes, you can see that as expected and as previously demonstrated, we have a progression from naive to stem cell memory, to central memory, to effector memory. And the way we read this graph, instead of having the typical one tab or one bar with a color code red for high or blue for low, that you will have in the bulk analysis, we are lining up here each cell of the cells that were sorted. So each bar that you see color-coded represents one individual cell within that gate of naive cells. So what this allows you to do is to basically, rather than look at the average expression, you can visualize at the single cell level how many cells within the naive T cell compartment express the gene of interest and at what level of expression. So it's an additional layer of information. And this becomes very important because while we confirm that we see the trend of naive effector genes expressed on the naive cells and effector genes like KLRB1 or granzyme K, granzyme A, you can observe that they progressively are expressed in the effector memory. It also informs us about the heterogeneity of the cells that we have sorted. So I really want to bring up the attention, for example, on the stem cell memory T cells. And while we see that LEF1, it's expressed fairly homogeneously by all the cells that we have sorted, if we look at other genes as we go down the list, you can see that only some cells express some genes. For example, if we look at CD58, you can see that there are 4 cells of all the cells that were sorted that express variable levels of CD58, of the gene encoding for CD58. Again, this strongly reinforces the concept that not all the cells that we've sorted within a gate are equal, and they have difference in the gene expression profile. Notably, you cannot find 2 cells that share the same profile, really emphasizing the need for a refined way to look at these cells and characterize these cells. And again, here I want to zoom on the total stem cell memory T cells. And on the left, I'm showing you the gate that was used to sort. So we sorted these cells as a total T stem cell memory based on double expression of CD95 and CXCR3. But if you remember from the introductory slide that I shared with you, at this point, if we did not use index sorting, we would be unable to say whether cells that express, a subset of cells express a gene of interest fall into the high end of the gate or the low end of the gate. And so the way we would do this conventionally, if we didn't have index sorting, would be to repeat our experiment. And now, instead of using a broad gate like the one that I show you here, sort CD95 high, for example, and CD95 low, if we want to see if there's a correlation between the level of expression of CD95 and a specific gene profile of interest. And that's exactly where index sorting comes in place, and it allows us to perform this kind of analysis without the need to repeat an experiment and change sorting gates. And that's because we have recorded the information of each cell that is being sorted. So now we can go back on Sigic and we can modify the gate the way we want. We are not locked down into the sorting gate that was used originally. We can change the gating because we have information for each cell at the protein and gene expression level. So instead of repeating the sort, we can change the gate, we can draw narrower gate, and in this case, we're analyzing on the left the 20 cells that express the highest level of CD95 and 20 cells that express the lowest level of CD95. And in real time, we can now recalculate the gene expression analysis, the differentiation gene expression analysis. And what we find is very interesting pattern. So if you look now at the right, if we look at the red arrows, that they are a representative of gene associated with an effector phenotype, we can see, for example, granzyme A on the top of the list, few cells expressing low level CD95 express granzyme A, and more express granzyme A when they acquire a higher expression of CD95. Similar for ARL4C, PEM129A, NKG7, and so forth. On the other end, if we look at naive express gene, we could, like FOXO1 or Actin1, you can see an opposite trend where these genes are more expressed by cells express low level of CD95 than high level CD95. Based on these observations, we can hypothesize that, for example, the progressive gain of CXCR3 and CD95 expression may mark the transition from naive to stem cell memory to central memory T cells, as, for example, illustrated in the first plot to the left by the arrow on total CD8 positive T cells, where we really hypothesize this transition delineated by the arrow. And now with the power of index sorting, what we can do, we can retrospectively go back to our data and further dissect all this population of interest to really validate our hypothesis. So you can see now on the plots from the blue to the red, we can slice up our original naive population into a population that has no CXCR3 expression to an intermediate CXCR3 expression. Similar things we can do now for the stem cell memory. We can dissect them into CD95 low and CD95 high. We can also appreciate that central memory, for example, could be further separated into CXCR3 positive and negative, but just for simplicity for this study, we'll focus only on further dissecting naive and central memory. And once we've done this and create this new gate, we can now repeat the differential gene expression analysis and interrogate these different subsets and correlate gene and protein expression. And so now here, these are the results where you can see that each column represents a specific subset of cells. At the bottom, we have the plot analysis with the corresponding protein expression. And we can correlate now the different level expression of CXCR3 and CD95 with different gene expression profiles. And observe, for example, that if we look at naive associated genes, like, for example, Actin1, in the bottom blue box, you can see that this gene is highly expressed on naive cells expressing no CXCR3, and it starts being decreased as the naive cells start expressing CXCR3. Interestingly, it's still expressed on the stem cell memory at CD95 low, but it's already lost, the expression of this gene is already lost when cells are transitioning to CD95 high and eventually central memory and effector memory. If we look at effector-associated genes, they're here in this red box. When we look at CD95 low stem cell memory and CD95 high stem cell memory, we can see a clear separation, and we can see that these genes are turned on only in correlation with high expression of CD95. So again, at the bottom we have the protein, on the top we have the gene expression, and we're able to correlate a more effector-like gene expression profile with higher level of protein expression CD95. So to summarize our observation, we started from a differentiation model based on protein expression only. But now, through the additional information that we've gained from the integration of protein and gene expression analysis mediated by index sorting, we have gained more information about different level of protein expression correlated with different gene expression profile. And we can hypothesize, for example, and refine intermediate stages of differentiation between the naive and stem cell memory T cells as described before. Of course, additional downstream function analysis or deeper gene expression analysis would be required to confirm this hypothesis. But another important consequence of this analysis is that now we have gained information about the correlation between protein expression and gene expression analysis, and we can refine our sorting gating strategy so that when we can repeat this experiment for downstream analysis, instead of using a broad gate as we used before, we can narrow down our gating based on the observation we just showed you to dissect even at the finer level the population of interest. In conclusion, what I showed you today is that integration of flow cytometry and single-cell RNA-seq allows for a deeper characterization of rare heterogeneous cell population. We also show that optimal panel design and single-cell sorting efficiently enable precise, targeted single-cell RNA-seq of only rare cells of interest. Furthermore, we show the correlation of protein and gene expression profiles that we were able to achieve through index sorting and novel bioinformatic tools led to the dissection and deeper definition of subset of T cells in different stages of differentiation. And of course, this was used as a model system just to emphasize these technologies and this approach, but the same approach can be utilized for a variety of heterogeneous biological systems. And finally, I would like to acknowledge all the scientists that have been involved in this project, in this team effort. From the R&D immunology application led by Suraj Saxena, I will particularly like to acknowledge Nihan Kara, the senior scientist who performed all the upstream flow cytometry analysis and single-cell sorting. And from the R&D genomic application led by Elaine Sham, I'd like to acknowledge Regina Lam, who performed the downstream single-cell RNA-seq. And I also like to acknowledge the FlowJo team with their support on data analysis, primarily Ian Taylor and then more. And thank you all for your attention. I would really like to take some time now to address all the questions you may have.